Lecture 10, where we will go over plant structure and function. There are three main tissue types present in plant organs: dermal, vascular, and ground tissue. Dermal tissue provides the outer protective covering. This typically has a waxy cuticle that will help prevent water loss. Vascular tissue is present to provide support as well as transfer materials throughout the plant, and ground tissue is important for storage, photosynthesis, support, as well as transport. These tissue systems are continuous throughout the plant and connect all organs to each other. Note the image to the right for the difference between the dermal, vascular, and ground tissue in the leaves, stems, as well as roots of the plant. There are three different types of dermal tissue that may be present in plants: the epidermis, cuticle, and periderm. The epidermis is considered to be the outermost layer of cells that covers the plant. It can be a single layer in non-woody plants or multiple layers in woody plants. The epidermis itself can be modified for various functions. It can be modified for gas exchange, as in the presence of guard cells. It can be modified for absorption in the roots, and it can also provide defense and prevent water loss on leaves. Trichomes may also be present to provide defense or to reflect excess light in high light environments. The cuticle is typically a waxy coating that is present on the surface of the epidermis. The cuticle is very important in preventing water loss and led to the success of plants in a terrestrial environment. The periderm is considered to be a protective tissue that is present in older stems and roots. The periderm typically replaces the epidermis in woody plants. Here's a look at some dermal adaptations that plants may possess. In the image to the left, we see guard cells that surround each stoma. Remember, a stoma is an opening that can help control water loss and gas exchange. In this instance, the guard cells can help control the opening and closing of the pores, further regulating gas exchange in leaves. Guard cells are present in all sporophytes except for liverworts. Trichomes are important in hindering the movement of pest insects. Trichomes can also be modified to secrete oils or chemicals that help in plant defense. When considering the vascular tissue of a plant, there are two different types: xylem and phloem. Xylem transports water and minerals in one direction from the roots upwards towards the shoots. Phloem can transport sugars produced by photosynthesis from leaves to other regions of the plant. Thus it can flow in both directions, up and down. A stele is a term that refers to the entire vascular tissue that is present in roots or stems. In angiosperms, the stele may form a central vascular cylinder in the roots, or it may form bundles in the stems and leaves. The ground tissue refers to all the remaining tissue that is not classified as dermal or vascular tissue. There are two different types of ground tissue in the plant: the pith and the cortex. The pith refers to the tissue that is internal to the vascular tissue. Note the picture below depicts the vascular tissue in purple. The pith is the yellow or ground tissue that exists within the bundle of vascular tissue. The cortex, on the other hand, refers to the ground tissue that lays external to the vascular tissue, as seen in the diagram below. During the development of the plant, cells become differentiated. This means that they have a specialized structure and function. We're now going to examine the different cell types that can exist in plants. These include parenchyma cells, collenchyma cells, sclerenchyma cells, as well as the conducting cells of xylem and the conducting cells of phloem. We're now going to take a minute to review the structure of the plant cell. Remember that the plant cell is surrounded by a cell wall. The cell wall is unique in plant cells and can include up to 3 layers. The primary cell wall is present in all plant cells. This is a thin and flexible layer that is produced during the phase of cell growth. The secondary cell wall, however, is not present in all plant cells. The secondary cell wall is a thicker layer that can lie underneath the primary cell wall. This wall typically is formed after the cell has completed growth. The middle lamellae is a thin extracellular layer that is comprised of pectins. This is important in helping adjacent plant cells to attach to each other. Please note the presence of the 3 different layers in the image to the right. We're now going to examine the specific plant cell types. First, we're going to examine the parenchyma cells. Parenchyma cells are present in leaf tissue as well as the fleshy tissue of the fruit. Parenchyma cells have thin primary cell walls and do not have secondary walls. These cells have a large central vacuole and contain various types of plastids, including chloroplasts and amyloplasts. These cells retain the ability to divide and differentiate into different cell types. Thus, they have a very important role in wound healing of the plant. This diagram here represents the parenchyma cells of a privet leaf. Note the large central vacuole that comprises most of the cellular space. Collenchyma cells have a thick, uneven primary cell wall. These are typically grouped into strands and located underneath the epidermis. These provide a lot of flexible support for the plant, but they don't restrain the growth in young shoots. At maturity, these cells continue to elongate in the stems and the leaves that they support. This diagram here to the lower right represents the collenchyma cells in a helianthus stem. The third cell type are the sclerenchyma cells. These have thick primary as well as secondary cell walls. These cells are very rigid due to the presence of lignin in the secondary wall. These cells are typically grouped into strands and located under the epidermis in regions that are no longer growing. Most sclerenchyma cells will die once they mature. When they die, they leave a rigid skeleton with the lignin behind and provide a lot of support for the plant. There are 2 different types of sclerenchyma cells, sclerids and fibers. Sclerids have a boxy, irregular shape, and they have extensive secondary walls. Fibers, on the other hand, are long and tapered cells and are usually grouped in strands. The fourth type of cell is the water-conducting cells that comprise the xylem vascular tissue. These cells are tubular, elongated cells that usually die and lignify once they reach maturity. This provides a conduit for water to flow within the plant. Secondary walls may be present, which are also lignified to provide additional support and also to prevent collapse as water moves through the plant. These are very typical in your woodier plants. The secondary cell wall is usually interrupted with pits or depressions where only the primary wall is present. These pits are important because they allow for lateral movement of water between adjacent cells in the plant. There are 2 different cells that make up the xylem, tracheids and vessel elements. Tracheids are present in all vascular plants. These are long, thin, tapered cells, and water primarily moves through the pits. Remember, the pits are the areas that just contain the primary cell wall. Xylem will also contain vessel elements. These are present in most angiosperms as well as some gymnosperms and seedless vascular plants. Vessel elements are short and wide, tapered and thin-walled cells. These cells arrange end to end to help form tubes. The extreme ends of these vessels have perforation plates. These will allow for water to flow throughout the tube. Note the difference between the 2 different water-conducting cells in the image to the right. The phloem consists of important sugar-conducting cells. Phloem primarily consists of long, narrow sieve cells. These sieve cells may join to form chain-like structures called sieve tube elements. These are responsible for carrying the sugar and other organic nutrients throughout the plant. The sieve tube elements usually lack a nucleus, ribosome, central vacuole, and cytoskeleton. This provides a lot of cellular space to allow for nutrients to flow through freely. These elements connect through sieve plates. These sieve plates usually have pores that will also help allow for nutrients to flow through. In addition, companion cells will run alongside these elements. These companion cells will connect via plasmodesmata, which is a type of cell junction. This image here depicts the important structures described on this previous slide. Note the presence of a sieve cell, which may join to form the sieve tube elements. Note the presence of a sieve plate in between the cells. Also note the cells have a tubular-like structure to allow for the passage of organic compounds. In the image to the right, upper right, we have the companion cells that lay alongside the sieve tubes. These connect into the sieve tube elements using the plasmodesmata.